Hello again, and welcome to Chapter 14. And again, we're entering new territory in this chapter. So let me see. So far, I've talked about induction, consolidation, and maintenance of LTP and memories. We've seen the roles of kinases and local mRNA translation and genomic cascades. We've seen the mechanisms of synaptic tagging and then ultimately spine stabilization, where short, stubby spines expressing the self-perpetuating superprotein kinase, kinase M zeta, are quite resistant to change. We've also seen how forgetting can happen back in, chapter 12, where small increases in the intracellular calcium concentration from ongoing neuronal activity can eventually break down the spine. Now imagine that many thousands or millions of spines are involved in the distributed network that stores the memory. The gradual removal of a spine here and there may initially have little impact on the memory, but the continued loss of these small components of the memory storage may add up to a significant decay, producing what we call graceful degradation. The memory may perhaps start to be a little more effortful to recall, and details are lost, but with effort, you can perhaps still call up the partially decayed memory and use it. However, today, we're going to explore those mechanisms of recall and reconstruction of a memory trace. You might be surprised at some of the amazing things that can happen to a memory in this process. I'm going to talk about traumatic events that can disrupt a memory, which we've seen a little bit previously. But I'm going to then use the evidence from those situations to illustrate what can happen in more subtle circumstances. I'm going to talk about animal experiments with fear conditioning. And we're going to take a quick look at active trace theory, not to be confused with active decay theory that we saw in Chapter 12 when we were looking at forgetting. And then we're actually going to spend a lot more time examining reconsolidation theory. And I think that's about as far as we'll get today. And I'll save the second half of Chapter 14, like Part B, to be delivered tomorrow. Once again, let's jump straight to the punchline. The main point that I'm going to try to convey in today's lecture is that memory that starts out labile and then stabilizes can become labile again. Let's just say that you have a memory for, some emotionally salient event. All the conditions have been met to encode that memory. The kinases were activated, the memory modulators were invoked, the mRNA was translated, and the genomic cascades were invoked, and then even the synapses were perforated and the spines perhaps were split to increase the synaptic communication between the neurons in the distributed networks that encode the memory. The storage of that memory remains labile during those processes of consolidation up to about 24 hours, until the newly synthesized proteins are all fully structured into the mature forms of the dendritic spines. After that, the spines are considered stable, and we typically think of them as resistant to change, undergoing only slow and gradual decline or graceful degradation. Now, all of that may be true, as long as that memory is kept in the background. However, if the memory is recalled, there's an abundance of evidence that the simple act of recalling the memory, or even reliving the situation wherein the memory was first formed, can cause it to become labile again. Now, the flowchart I drew on this slide shows that new memories are labile. But through the process of consolidation, they then become stable. Now, if some stimulus causes the memory to be recalled, or actually reactivates the memory by exposing the person or animal to the same or similar stimulus situations, then the memory becomes labile again. And there's a new process of reconsolidation, during which time the memory is once again vulnerable until it is reconsolidated. So I hope that by the end of today's lecture, you're going to have a good idea about these processes, and I will have convinced you that the reenactment of a stimulus event is actually going to cause great restructuring in the spines that originally stored the memory event. I'm going to remind you that back in chapter 11, I showed you that traumatic events, including electroconvulsive therapy, disrupt memory events that are still undergoing consolidation. In other words, new memories are vulnerable. Back in the 1960s, it was thought that only new memories, like things that happened that morning, were vulnerable. So electroconvulsive therapy, or ECT, was thought to be a very, very safe procedure, and it was actually used quite routinely. Now, the images on this slide are actually from a movie from 1975. Jack Nicholson starred in One Flew Over the Cuckoo's Nest, based upon the 1962 novel by the same name, written by Ken Kesey. The book is actually a literary classic now, but in 1962, it was banned in many states for being too contrary to social norms and etiquette. Imagine that. Anyway, Nurse Ratched was the evil overseer of the psychiatric ward, and Jack Nicholson was the fun-loving, totally out of control, wacko patient, Randall McMurphy, who wreaked havoc in the ward. I won't tell you all the details of the movie, but it's an interesting, although not quite scientifically accurate, depiction of the use of ECT and psychosurgical procedures back then. Now, the movie may be a little dated now, but I still recommend it. And anyway, what am I doing? This isn't movie appreciation class, right? I really just stuck these images here to remind me to talk to you about how labile memory is in the early stages, and that back then, when everyone thought ECT was safe. But now, with the knowledge that we have about how long-term memories are not invulnerable, the decision to use ECT, I think, must be handled much more conservatively. Early evidence of the potential detrimental effect of electroconvulsive therapy came from Missinen and collaborators in 1968. They partially deprived rats of water, so they were made thirsty, and in the process of giving limited access to water, they trained them to get their water by licking a spout in a lickometer. This is a highly sensitive device that can measure the very, very rapid licking behavior of rats when they're licking at a spout. So they allowed the rats to lick 110 times, and then the spout was withdrawn. So this was the procedure for days 1 and 2. And then on day 3, they divided the rats into 3 groups. All 3 groups were given the same cued fear conditioning with the standard kind of tone plus foot shock experimental design that we've seen so many times before. But then after the conditioning session, group 1 got immediate electroconvulsive shock, whereas groups 2 and 3 were just returned to their home cages with no electroconvulsive shock. Now we know that ECS disrupts the recent memory, so hopefully everyone's figuring out that group 1 that got ECS represents a group in which the consolidation of the fear association is disrupted. They should hypothetically have no memory of the tone foot shock association. The other two groups that were returned to their home cages would nicely consolidate their tone foot shock conditioning. Okay, then on day 4, all 3 groups were returned to the chamber and given a single presentation of the tone, a single presentation of the CS, with no foot shock, with no US. So this served as a reminder, I suppose, of the fear conditioning. On this day, groups 1 and 3 were returned to their home cages, but group 2 was treated with electroconvulsive shock immediately after the session. And let's move on to day 5. On day 5, all groups were returned to the lichometer environment. However, this time, the CS, the tone, was presented after each rat did 100 licks. After the presentation of the CS, instead of measuring freezing, as we've seen before, they measured the disruption of the licks at the water spout. Hypothetically, if the animal remembers the association between the tone and the foot shock, when the tone is presented, the animal is going to stop licking at the water spout. Now, if you were really processing the description of the experiment and thinking about it in relation to the information I've previously given you about how electroconvulsive shock disrupts memory consolidation, and also that recall or retrieval of a memory makes it labile again, then you're likely able to predict the outcome of the experiment. But of course, this was big news when this experiment was reported back in 1968, when they were pretty freely administering electroconvulsive therapy to all kinds of different patients. Okay, so let's see. And I apologize for the kind of shaky appearance of this graph, but it is from 1968, remember. Okay, so let's see what happened. In group 1, the fear conditioning was disrupted by the electroconvulsive shock that was administered after the conditioning session. So they continued to lick during and after the presentation of the tone on the last day of the experiment, on day 5. In group 2, the memory of the fear conditioning was retrieved by experiencing the same stimulus on the day after training, and then that newly labile memory was disrupted by the electroconvulsive shock after that reminder session. And so they also continued to lick during and after the presentation of the tone on the last day of the experiment. And then group 3. The fear conditioning was intact because there was no electroconvulsive shock after conditioning or after the additional presentation of the conditioned stimulus. So they actually stopped licking during and after the presentation of the tone on the last day of the experiment. So this experiment, of course, revealed that retrieved memory is newly vulnerable again. And of course, that called into question the idea that ECT, electroconvulsive therapy in humans, would only disrupt new memories from the time right around the administration of the ECT. What if the patient was thinking about other memories at the time of the ECT? well, those memories might become vulnerable and subject to elimination by the ECT. So, is everyone convinced that recalling or retrieving a memory can cause that memory to become more vulnerable? Maybe, maybe not. All right, let's convince you with another experiment. But I have a problem here. I have fumbled and bumbled over some of the names of scientists in my lectures so far. But the good news is that it shows that neuroscience is a very healthy and vibrant international community that's continuing to incorporate more and more diverse and underrepresented groups, and to foster science funding all over the world. We still have a long way to go, but if you look at the website for the Society for Neuroscience, you'll find a mission statement with a very strong commitment toward these goals. But the bad news is that I cannot even imagine how to pronounce the name of the first author on this study. I'll do my best and try Przybyszlowski. Okay, Przybyszlowski and Sarah. My apologies for the pronunciation, but in 1997, they trained rats on the radial arm maze, where the maze was consistently baited in 3 arms, as shown on the diagram. In every trial, the same 3 arms were baited, and they actually baited the arms with Kellogg's Cocoa Pops, or Cocoa Puffs. So you can imagine that this is a really tasty reward that rats will really work for. Now, in this experiment, they overtrained the rats so that the rats made no more than 1 error across 3 consecutive days. That is, they never entered an arm that wasn't one of the consistently baited arms, and they never entered an arm that they had already got the bait from. So no more than 1 of those errors across 3 consecutive days. The rats were actually trained for up to 40 days until they reached that criterion. So then at the end of the session, when the particular session, when any individual rat reached the criterion, the rat was injected with the NMDA receptor antagonist MK801, either after 5 minutes, 60 minutes, 180, 240, or 300 minutes. And then 24 hours later, each rat was tested in the radial arm maze with no drug treatment. And you should be able to see on the graph that when MK801 was administered 4 or 5 hours after the last training session, it had no impact. The rats continued to perform with an average of less than 1 error on this trial. But if the MK801 was administered during the first 3 hours after this final training session, the mice were impaired, or the rats were impaired, making more errors on the radial arm maze than they had previously been doing. Let's think about what this means. The rats trained daily for up to 40 days on the radial arm maze. At first, their behavior would have been very erratic, exploring arms randomly. But after dozens of sessions, they slowly make fewer and fewer errors. Now, in these daily trials, they're rehearsing the learning again and again and again. And when the experimenters brought them to a level of learning that was consistently high and well established, the administration of a drug could disrupt the memory, but only if administered during the consolidation period after a trial. the implication is that memory is an ongoing process that is constantly undergoing reorganization. Each and every time you rehearse your piano for the upcoming recital, or practice your lines for the school play, you are rendering the learning vulnerable. And if there's a disrupting influence during that consolidation period after that rehearsal, like there was in the rats in this experiment, your memory can be impaired. Now don't worry, because most of us are not going to undergo ECT or take a shot of MK801 after playing our recital piece. MK801 is actually psychotomimetic in humans anyway, so not much risk that you would want to go and take it. However, in light of this evidence, I would not recommend that you go out and get drunk after reviewing your neuroscience notes. Not that I'd recommend going out and getting drunk anyway, right? So, I don't know about you, but I'm pretty convinced that recall or retrieval of a memory puts it into a vulnerable state. The next question is, how and why does this happen? And as I said that, I realized, okay, technically that's two questions, how and why. But here's one explanation, called active trace theory. And fair warning, I'm going to describe another theory starting on the next slide, one that I think is far superior. But let's look at active trace theory. This theory, not to be confused with active decay theory of forgetting, which we saw previously, contends that there are two ways in which a memory can be in a vulnerable state. The first case is simply that the memory is new, and therefore it's in an active state. We've seen lots of instances where new LTP or new memory is actually vulnerable for disruption, so this makes sense. The second case is when retrieval cues are presented and a well-preserved memory is called back into a vulnerable state. In both of these situations, the focus is on the idea that the memory is now active. And if it's active, then it's thought to be vulnerable. When it's consolidated into the long-term storage, it's thought to be stable. But even for a stable memory, if it's called back up into the active form, that active form can then interfere with the stably expressed memory. Now, I find this theory unsatisfactory because although it generally fits with the data we've reviewed so far, it doesn't actually explain the mechanism whereby an active memory undoes a stable long-term internal representation. At its core, active trace theory contends that the amnestic agent, that electroconvulsive shock, or the trauma, or the drug, is actually the cause of the amnesia. All right, so we're next going to talk much more extensively about reconsolidation theory, the perspective that I favor. In reconsolidation theory, the trauma is not the cause of the amnesia in the case that a memory is disrupted. The newness of the memory, or the reactivation of the memory, is going to be the cause of the amnesia. The amnestic agent, the electroconvulsive shock or the trauma or the drug treatment, is simply blocking the reactivation of a synapse that was disrupted by the reactivation of the memory itself in the case of reconsolidation. So let's move on to reconsolidation theory, which proposes that a recalled memory causes the structures that underlie the memory to be partially broken down and then rebuilt. So maybe the best way I can show you reconsolidation theory is to illustrate the underlying principles. First, we know that a new memory is vulnerable to disruption if a traumatic event or a drug like, well, let's say the translation inhibitor anisomycin is injected during the consolidation period. This slide actually shows the timeline of a standard experiment with CSUS training, followed by an injection of anisomycin into, let's say, the basolateral amygdala at the start of the consolidation period. Later, when the rat or mouse is presented with the CS, it doesn't freeze because the memory consolidation was disrupted when the spines could not translate available messenger RNA, or when the genomic cascade supplying new messenger RNAs was interrupted by the injection of anisomycin. But we also know that the disrupting factor has to be administered during the consolidation period. If anisomycin is injected after the consolidation, the memory appears less vulnerable. And this lower timeline of the experiment, if we do it this way, then when the rat is tested, the memory of the CSUS association is intact, and the rat freezes when presented with the CS tone. So those first two descriptions of experiments were fundamentally experiments with new memories. And if anisomycin was injected at the time of consolidation, then the memory is disrupted. If anisomycin is injected after the consolidation period, then the memory will consolidate and will be preserved. So those are experiments with new memories. But in this third version of the experiment, let's see. The rat is trained in the cued fear conditioning. And then the memory, the consolidation period is uninterrupted. And then after, let's say, 24 hours, the rat is tested. And it shows the freezing response when the conditioned stimulus is administered. But now, after the test, the rat is injected with anisomycin during the reconsolidation period. And then when tested 24 hours later, it doesn't freeze as much as it did in the first test. The simple presentation of the retrieval cue, the CS, at test number 1 actually made the memory vulnerable. And the idea of reconsolidation theory is that retrieval or recall of the memory means that it has to then reconsolidate, and disruption of the mechanisms that underlie that the initial consolidation in the first instance would also disrupt reconsolidation in the second instance. So let's look at an actual experiment. In the year 2000, Karim Nader, a postdoc in Joseph Ledoux's lab, performed an experiment much like the one I outlined on the previous slide. They conditioned the rats in the cued fear conditioning on day 1. They allowed uninterrupted consolidation and then tested them on day 2, labeled as test 1 on the timeline. And then you can see the upper graph shows that all the animals, remember they haven't had any treatment yet, just the fear conditioning. So all the animals show little freezing behavior prior to the CS presentation, and lots of freezing behavior after the CS presentation. But the rats were then injected into the basolateral amygdala with either artificial cerebrospinal fluid or one of 2 doses of anisomycin. The lower graph shows that the rats injected with the artificial CSF or the low dose of anisomycin showed high levels of freezing in this second test. But interestingly, the rats injected with the high dose of anisomycin seem to have lost the association. They show little freezing after presentation of the tone in all the trials of test number 2. If you remember, when I talked about anisomycin long ago, well, at least a week ago, I described that you have to give a high dose and knock out most of the protein synthesis in order to disrupt consolidation. So bravo for Nader and associates. They demonstrated that disruption of reconsolidation under conditions that would produce substantial protein synthesis inhibition impaired expression of the previously consolidated memory. But they also demonstrated that anisomycin injected 6 hours after test number 1, that is, after the reconsolidation period, failed to disrupt memory of the CS-US association. Once again, that you can see that in test 1, all rats showed freezing behavior after presentation of the CS tone. And then at the second test, whether the rats were injected with artificial CSF or high-dose anisomycin after the consolidation period, well, the rats all show high levels of freezing behavior. Now, this really strongly supports the idea that reconsolidation is a protein synthesis dependent restructuring that follows the same kind of timeline as the original consolidation of the learning. If the anisomycin was given in the reconsolidation period, it disrupted that reconsolidation. And if given after that standard period of time, that's the same for reconsolidation as it is for consolidation, then if it's given that late, it's not effective. It doesn't block the reconsolidation. But they weren't done yet. They also administered the artificial cerebrospinal fluid, or anisomycin, into the basolateral amygdala without doing test number 1. So it's at the same time as in the first of these experiments that I showed you. And then they tested 24 hours later. Now, in this experiment, there's no graph number 1, there's no upper graph, because there's no test number 1. But in the lower graph, you can see that the anisomycin did not disrupt the ongoing original consolidated memory of the CS-US association. So you can see that the anisomycin treated and the artificial CSF treated rats all exhibited high levels of freezing in this test. So it's not some non-specific effect of the anisomycin. It actually is the reactivation of the memory and the requirement for reconsolidation is absolutely essential for this effect of anisomycin. And in the last of their series of experiments, they again trained the rats in cued fear conditioning and tested, but this time there was a 14-day delay between the original CSUS training and test number 1. Now, the top graph shows that the rats once again exhibited very little freezing prior to the CS presentation and robust freezing after the CS presentation. Now, anisomycin was injected after test 1, and the lower graph shows that the anisomycin once again dramatically disrupted reconsolidation. So the rats showed little freezing even when the memory had lain dormant for 14 days prior to reactivation. Now, I hope everyone is fully convinced that every time you re-engage a memory, you're going to remodel it in some way, and that the remodeling looks very similar to the initial consolidation process. Okay, so I recruited Wreck-It Ralph to help me make this point. Active trace theory proposes simply that when a memory is called into the active state, it's vulnerable. Now, reconsolidation theory proposes that when you reactivate or recall a memory, there must be some smashing of the original encoding of the memory. You actually tear down aspects of the internal representation, and then you have to rebuild them, perhaps stronger, using the same mechanisms that were used to consolidate the original memory. All right, so maybe we should put Wreck-It Ralph away before he smashes the internet again, because after all, this is an online course. But wait, here I go asserting that the same mechanisms are invoked as in the original consolidation, just because by blocking consolidation mechanisms, we block reconsolidation. But are they in the same spines? Are you actually smashing up the internal representation by modifying the same spines? Well, Valérie Doyer and colleagues in Joseph Ledoux's lab examined this with another one of those, I think, really smart experimental designs. In this case, what they did was they trained the animals in cued fear conditioning with 2 CSs, 2 conditioned stimuli. They called these CSR and CSN, where CSR means that this CS was reactivated, and CSN means that this CS was not reactivated. So on day 1, they exposed the rats to the 2 CSUS pairings. One pairing was a pure 1 kilohertz tone with foot shock. The other pairing was a complex frequency modulated tone, which is a combination of a variety of different frequencies, also paired with foot shock. The rats were conditioned and then allowed to consolidate without any disruption in between. And then on day 2, 24 hours after the initial training, the rats were injected with either U0126 or vehicle into the lateral amygdala. Now, U0126 is a MAP kinase inhibitor. It's especially effective in blocking signaling through CREB to prevent expression of the immediate early genes CFOS and CJUN that we know dimerize to interact with the AP1 response elements. We saw all of this, quite a few chapters ago when we looked at CREB. So accordingly, we would expect that U0126 would disrupt consolidation, or in this case, reconsolidation, by disrupting the genomic cascade. So, 30 minutes after administration of the MAP kinase inhibitor, the CSR was reactivated with a single presentation of the tone. Now, of course, to keep things balanced, for half the rats, the CSR was the simple 1 kilohertz tone, and for the other half, it was the frequency modulated tone. Now, Doyer and colleagues tested all the rats 3 hours after the CSR reactivation to test short-term memory. You can see in the top graph that all the rats responded by freezing to the additional presentation of the CSR. But then Doyer and colleagues tested the animals again on day 2. And this time, they found that the rats that were injected into the amygdala with vehicle had robust freezing responses to both the CSN and the CSR. There might have been a small reduction in the response to the CSR as it had been presented twice without shock, but this wasn't a significant effect. On the other hand, the rats that were injected with the MAP kinase inhibitor showed robust responses to the CSN and impaired memory for the CSR. The obvious conclusion is that interrupting the reconsolidation process was very specific for the stimulus that was reactivated at the time when the pharmacological manipulation was delivered. The MAP kinase inhibitor blocked reconsolidation of the reactivated stimulus and had no impact on the ongoing internal representation of the CSN stimulus association that was learned at the same time as the disrupted CSR stimulus association, but was, shall we say, laying dormant at the time of the pharmacological manipulation. And here's another interesting variation on the reactivation paradigm. In this experiment, Ben Mamou and colleagues in Karim Nader's lab investigated the mechanisms that contribute to destabilization of the memory during reconsolidation. And at this point, I also have to note that it's nice to see that Karim Nader, who was the postdoc in charge of one of the earlier experiments from Joseph Ledoux's lab, was now running his own lab and directing some cutting-edge science. But, well, back to the experiment. Nader's group conditioned rats in standard cued fear conditioning and allowed uninterrupted consolidation. Then, 24 hours after the cued fear conditioning, they gave an injection of ifenopril into the basolateral amygdala, and then they reactivated the CS, and then they injected anisomycin. Or they reactivated the CS, gave an injection of ifenopril, and then gave an injection of anisomycin. Now, the order of these events is very important. You see, ifenopril is an NMDA receptor antagonist. If it's injected before the CS, it would hypothetically prevent the plasticity-inducing actions of the CS. But if injected after the CS, it's basically a control, allowing the CS to have its impact on the spines. And then in either case, we can inject anisomycin to disrupt any reconsolidation in both cases. So, in experiment 1, they injected either vehicle and then vehicle, or they injected vehicle and then anisomycin, or they injected ifenapril and then vehicle, or they injected ifenapril followed by anisomycin, with a reactivation of the CS between the injections. Now, all the rats were tested for memory of the conditioning shortly after the anisomycin injection at a time point when the memory trace would not yet be dependent on the protein synthesis. And so you can see that all the rats froze in response to the CS presentation. And then the rats injected with 2 vehicle injections reconsolidated nicely and had robust freezing on test 2 the next day. The rats that were injected with vehicle plus anisomycin showed that the anisomycin disrupted reconsolidation, and the rats did not freeze in test 2, as you might expect. We know that anisomycin disrupts reconsolidation. And the rats that were injected with ifenapril followed by vehicle continued to freeze. There's hypothetically no reconsolidation process because the ifenapril blocked the NMDA receptors. But the interesting group is the group treated with ifenapril plus anisomycin. In this case, the ifenapril blocked the disrupting effect of anisomycin. In other words, blocking the NMDA receptors prevented the whole cascade of reconsolidation. So blocking protein synthesis with anisomycin couldn't disrupt the memory because the cascade of tearing down the spine structures, like breaking up the actin and all the subsequent steps, did not occur. There was no Reckitt-Ralph effect and nothing to actually reconsolidate. So the anisomycin was ineffective. And then in experiment 2, the major difference was simply that the ifenapril was given after the influx of calcium happened. So in this case, the vehicle plus anisomycin, those animals, and the ifenapril plus anisomycin, these animals, all displayed the effects of anisomycin, preventing the reorganization of the synapses because the presentation of the CS reactivation caused that initial tearing down of the structures. And the protein synthesis cascades that anisomycin was then blocking were impaired by the injection of anisomycin. And you might guess that other mechanisms that are invoked in consolidation are also invoked in reconsolidation. But here's some evidence to verify those suspicions. We know that plasticity-inducing synaptic activity through NMDA receptors and voltage-gated calcium channels activates the ubiquitin proteasome system to remove some proteins to allow spine reorganization to occur. In the timeline at the bottom left, you can see that Lee and colleagues exposed the mice in their experiment to contextual fear conditioning. Not cued fear conditioning this time, but contextual fear conditioning. And some mice some controls, had no conditioning. And then they exposed the mice to the context again on day 2. 15 or 30 minutes later, they dissected the hippocampus and used an assay to pull down the total polyubiquitinated proteins from the dissected hippocampi. The assay shows that in comparison to naive mice and mice that did not have the CS reactivation, that then 15 minutes or 60 minutes after reactivation, the mice have elevated levels of polyubiquitinated proteins in the hippocampus, suggesting that the mice are engaging in one of the consolidation activities we saw previously, breaking down proteins to limit the extent of synaptic potentiation and to remove transcriptional repressors that would keep the genomic cascade suppressed in between plasticity events. They also showed that blocking the ubiquitin proteasome system prevents destabilization of the trace. In this experiment, the mice were conditioned and allowed to consolidate for 24 hours. They were then injected into the hippocampus with vehicle or the proteasome inhibitor beta-lac or anisomycin or beta-lac plus anisomycin. The vehicle-treated mice showed intact reconsolidation, of course, and exhibit robust freezing on test 2. The beta-lac injected mice also exhibit robust freezing because the proteasome was blocked, so hypothetically there was never any reorganization of the spine. And, well, the anisomycin-treated mice, of course, lost the contextual fear association, so they did very little freezing when placed back into the shocked context on day 2. And again, the most interesting effect is the drug interaction. The beta-lac blocked the disrupting effect of anisomycin because apparently scaffolding proteins are not broken down, and then anisomycin cannot have any effect. Without breaking down the scaffolding proteins, the entire reconsolidating process is blocked. It's as if the animals had not been exposed to the contextual conditioned stimulus reactivation. And still, Lee and colleagues weren't done. The last experiment in their science paper was an analysis of specific proteins that were being trafficked off to the proteasome. The graphs at the right show that polyubiquitinated versions of the scaffolding proteins SHANK and GCAP, or guanylate kinase associated protein, were elevated 60 minutes after contextual CS activation. They also show that total SHANK protein was decreased over the first 2 hours after reactivation and then recovered by 6 hours and 24 hours. And these data are also shown in the Western blot above the graph. So polyubiquitinated SHANK and GCAP increase, and the total amount of SHANK that is available in the spines decreases over the first couple of hours and then rebounds back afterward. I hope everybody is thinking that way back when we talked about initial consolidation, I described exactly those kinds of processes, that we break down the cytoskeletal structure and the postsynaptic density proteins like Shank, and then we build them back up again. So it looks again like exactly what was happening in the original consolidation is happening once again in reconsolidation, after all we do is present a reminder, a retrieval cue of the initial memory event. Now I've shown you evidence for reconsolidation from cued and contextual conditioning experiments. And there are additional published reports that have described manipulations in inhibitory avoidance experiments. And it wouldn't surprise me if there are additional experimental paradigms that I haven't come across that have been found to demonstrate the effects of memory reconsolidation. Now, this slide also shows you a sampling of the many mechanisms that have been found to play a role in memory reconsolidation. And if most members of the list look a little bit familiar, well, they should, because these are kind of all the same mechanisms that have been implicated in memory consolidation in the first place. So we have now seen a wealth of evidence showing that when a memory is reactivated, the initial steps are steps that actually weaken the synapses underlying the memory. There's calcium influx through NMDA receptors and voltage-gated calcium channels. Kinases are activated. The protein scaffold is polyubiquitinated and broken down by the proteasome, and AMPA receptors are actually removed. If we block these steps from occurring, the reconsolidation effect is blocked or diminished, and reactivation of a memory appears to be without consequence. However, if we do not intervene, the synapses are at first weakened, but then potentiated again. And we all know that repeated practice can make for much stronger associations and much stronger skills. So hypothetically, the reactivation ultimately lends itself to further strengthening of synapses and enhanced learning. But if we block those subsequent steps, like with anisomycin, we're left with a broken down synapse that fails to be restructured into a new and functional potentiated member of a complex circuit encoding a strengthened memory, perhaps with additional multiple spine profiles and stable, short, thick, mature spines. Now, I also want to remind everyone that these events occur in circuits that underlie either indexing or storage of memories. Whenever I talk about changes occurring in spines, we need to think beyond that and understand that the changes are laid down in many thousands of spines to encode any individual memory event, or by inference, the reconsolidation of any memory event. So, the concert pianist who practices day after day after day after day. Think about it. The circuits that are encoding that skill are actually being modified with each session of practice, being broken down and reconsolidated over and over and over again. Or at least from our biochemical assays and our current sparse knowledge about how the actual memory functions are laid down in the brain, it looks like this is the kind of mechanism that's happening. And I think I'd better stop there, and we will continue this tomorrow with chapter 14b. I've got some really cool stuff to tell you about memory reconsolidation in 14b. We'll see you then.